first major differentiation that we have to make in this chapter is the difference between chromatin and chromosomes. Chromatin is made up 40% of DNA and 60% of protein. So at any given time, the majority of the time during the cell cycle, you have DNA in the form of chromatin, not in the form of chromosomes. Chromosomes are only present during actual cellular division, so during mitosis, during meiosis. Otherwise, the cell will have DNA in the form of chromatin. Now, the chromatin, there's two major types. You have heterochromatin and euchromatin. Heterochromatin and euchromatin. So with heterochromatin, they're chromatin that are not expressed, okay, at that one point in time, while the euchromatin are expressed at that one point in time. Okay, expressed as far as actively undergoing the process of gene expression, and which we will talk about ad nauseam in the upcoming weeks. Okay. Now, as far as actual chromosome structure, here we have examples of chromosomes, an actual depiction of the chromosomes using a particular staining method. So, here we have the diagram figure 10.5. So in figure 10.5, we're able to see the basic structure of our chromosome. So over here we have the chromosome as we typically see it in its X-shaped formation. In the middle, we have the holding it together a protein that makes up the centromere. Okay, different from what we actually consider the centriole. Centromere, centriole. The centriole goes to opposite poles and makes the spindle fibers to allow for mitosis to occur, while the centromere is there basically to connect our sister chromatids together. The sister chromatids are the branches of these, or the arms of these chromosomes that actually connect the sister chromatids together. The centromeres connect the sister chromatids together. Okay, as you can see here, these would be the sister chromatids, the arms of the chromosome. Now, when we're looking here, basically the chromosome, once it's in its condensed form, it shows up as a basic scaffold protein in the middle, and you have your rosettes, okay? Basically looking like a flower structure, thus the name rosettes. Now, in each one of these rosettes, we have several chromatin loops, okay? And it's just a loop structure of chromatin, essentially, okay? Just really highly condensed chromatin, and that's basically what a chromosome is. It's highly condensed chromatin, and this DNA is highly condensed around structures called histones, okay? So as we can see here, chromatin loops are made out of these structures called solenoids. Okay, these solenoids are highly condensed, essentially nucleosomes, and as we can see here, this is a nucleosome. The nucleosome is a histone core, which is wrapped around by our basic DNA structure. And of course the DNA is in the basic duplex form of the DNA double helix. Okay? So as you go through this question on the workshop, you can go from the top of the hierarchy all the way down to the bottom, and to review, you go back up. So the DNA double helix, which is wrapped around the core of histones, and there are multiple histones that are involved in this complex that we call the nucleosome. So we have the complex of the histone wrapped around the histone. We have the DNA, and that makes the nucleosome. A bunch of nucleosomes, as we can see here, makes up the solenoid. A bunch of solenoids will wrap around in a loop to make the chromatin loop. A bunch of these chromatin loops will come together and make the rosettes, and the rosettes will all come together to make the chromosomes. Okay, there's a particular protein that's mentioned that actually aids in the process of the condensation of these of this chromatin structure, and that is the protein called condensin. Okay. Now we will talk about. Well, we might as well talk about it now. So we have the concept of the karyotype. As we karyotype is basically just a map of our chromosomes. As you can see here, we have our 46 chromosomes. That's our diploid number. Our haploid number would be 23 chromosomes. So we have 23 sets of chromosomes, basically aligned in order by size, one being the largest and 22 being the smallest, essentially. Okay? With our sex chromosomes making up our 23rd set of chromosomes, either XY or XX, depending on whether we're males or females, respectively. So the way that we attain these karyotypes is by doing tests such as amniocentesis and chorionic villi testing, which we talked about actually next week in our workshop involving, I believe it's chapter either 12 or 13, I don't remember which chapter it's in. So you perform these tests on a pregnant woman to be able to tell if the child has any genetic abnormalities as far as the number of chromosomes, so whether they have trisomies or monosomies, which all fall under the umbrella of diseases of aneuploidy diseases, okay, which are, and an aneuploidy is a disease caused by an abnormal number of chromosomes, okay, such as Down syndrome, Klinefelter's, etc. And we will see that in the next chapter as well. So you can introduce this basic concept of the karyotype, but we will come back to it as far as using it as a tool to understand aneuploidies next week, okay? But they should know at this point what the karyotype is, okay? So just as a review of terms, we can see in this figure 10.7, we have homologous chromosomes, okay? And these homologous chromosomes are not connected via the centromere. Homologous chromosomes are not connected via the centromeres, while the sister chromatids are connected via the centromeres. Now, the other thing that this particular diagram is showing is how the chromosomes essentially would look if if they were condensed during the cell cycle before replication and immediately after replication, if they were condensed. Okay. So after we end the process of mitosis of cellular division, we go back into the G1 phase. And so with the G1 phase, we essentially have chromosomes that look like this. Okay, not the typical X structure that we think of when we think of chromosomes. It isn't until after DNA replication, the S phase, that we actually get the X structures that we typically think of. Okay, because this chromosome, this part of the chromosome that we see here, basically this one here, okay, this one was copied during the S phase to make the other strand, the second strand. Same concept applies here. We started off with this one, and after replication, we are able to replicate the other side, okay, as we can see here, okay, and form the typical X shape of our chromosome, okay. And we'll talk about a couple more specifics as far as the cell cycle in the next video.